Component 3 in-class questions. Question 6. Plant transport. Bit of a weird question is this. I bet you're glad you didn't get this on your real exam. The apparatus is set up to study upward transport of mineral ions from the soil. So the key there is upward transport of mineral ions from the soil. And we're using this mineral ion. So this is just a radioactive potassium. So it's just like normal potassium in every way apart from it's radioactive, so we can detect it. Now, you might be using a Geiger counter or some other way. And we can see the potassium is starting there in the soil, and then it's presumably moving up there, and we're testing it there. And we've got xylem. There's our xylem. And then we've got phloem on the outside of that. And there's our barrier. So that's our phloem, and then we've got our xylem. So what do you already know about or think about? We think about xylem contains water, so water going up from the roots to other parts and then being transpired out of the leaves, and minerals dissolved in them. So that's kind of what we already know. And then when we look at the results, we can see below the point, so this is kind of this area here is below equal, and then above, there we go, equal again, and this is the middle bit where the phloem doesn't seem to have any in. What happens to it? Who knows? Maybe it gets used in some sort of pump. We usually have sodium potassium pumps, and maybe it goes down for that reason. But we don't know. It gets used. But the xylem is actually double the other values. Okay. So you might think that, from what we already know, that the potassium goes up in the xylem. Okay. So that's our first mark really. So K plus transported upwards in the xylem. And then what happens? Well, the potassium moves from the xylem to the phloem, and down here we can see how that's possible. Up at the top we can see how that's possible. Where there's a barrier, that doesn't happen, so we don't get any transfer, and that's why if it's been used in the phloem, then it would go down. So the potassium only moves sideways or laterally from xylem to phloem. Does the potassium move along the phloem with the flow either up or down? Well, there's no evidence to suggest that. I'm going to ignore that 11 as a minor point, but from below, well, does it go upwards? Well, it doesn't seem to be the case. If it moved in the phloem, then that number would be higher. So the K plus does not move up or down in the phloem. It's a little bit of a strange experiment, and part 2's equally strange. The control for this experiment it's saying is don't use that waxy barrier. You can separate them, but don't put the barrier between. So presumably they would still have a kind of watery sort of tissue connection. Why would this be a suitable control? And explain how the results could be used to support the conclusion. So 2 parts here. So why would this be a suitable control? Well, remember what a control is. It's to run the experiment to show that the thing that we're changing, so this barrier, is the thing that's having the effect. So that's really what we need to say for the first one, because that's the actual thing that we're testing there. So to show that the wax paper stops the movement. And we don't get a lot of space to write this. To show that the waxy paper stops the movement. And then how the results could be used to support your conclusion. Well, you could do a few things here. You could say that if you put them back into contact again after you separate them, so you separate them, put them back into contact, then you'd expect movement to carry on, and the values would be the same. So for example, if separated then returned, then expect values to be the same as in the original experiment. So remove this waxy barrier, but just separate them. Maybe the act of separating them into something, so you put them back and you'd expect results. You'd expect the transfer across. Or if you keep them separated, then you'd expect these values here. So if you keep them separated, you'd expect this experiment. If you put them back together again, you'd expect these numbers in the phloem to be high. Weird question. Sorry. And the weirdness continues. Part B. And you might have seen similar experiments using a bag of radioactive CO2, and they're also using some radioactive phosphorus in phosphate, and the same waxy strip situation there. What conclusions can be made from these results about the transport of 14C in xylem and phloem? Well, we can certainly make a statement about the result of 14C in xylem. Well, there's none. So we can definitely say no transport in xylem. No transport of 14C in xylem. And for the second mark, the second mark is the transport in the phloem is in both directions. How do we show that? Well, we can see transport in phloem of the carbon going 1, 2, 3, 4 down there, so we can see it going, that's certainly going down in that direction. We could guess from the phosphate or the phosphorus results, which go the other way, so phosphorus coming in there and then seemingly travel upwards, that the carbon would be able to do that as well. It's not conclusive, but that seems to be what they're getting at there. So these results as a whole would show that the phloem moves in both directions. So phloem moves in both directions, and that would tie in with what you know from the theory. So you're always looking to put your theory points into answers, and you know that phloem transport is bi-directional. Then part 2, describe how you would refine the experiment to determine the direction of transport of phosphorus. Then you would suggest, well, I've checked it going upwards, so I'm going to check it going downwards now. So I'd check it under the leaf, and then if it's gone both ways, then we're happy that it's going in both directions. So 2 marks here. Analyze samples, below the leaf also. So we've done it above the leaf, so try below the leaf. Then if positive, or you get some numbers there, then movement is in both directions. So a little bit more straightforward with that one. And then thank goodness for part C. Part C is just, here we go. Here's a chance to give some of our knowledge. So we know what sources and sinks are. A source of sugars is the leaves in the summer, and the sink is the tubers, the potatoes. And then in the spring, the source of the sugar is the potatoes, and where is it being used? The sink. It's those stems and leaves that are growing, so they're respiring. The question wants, use the mass flow model. So that's our bit of theory to explain how the sugars are transported in spring. So we're only really talking about this area here. We could say where the sugars have come from, but this is the only kind of bit of transport that we're actually using. So we could set this up by saying in summer the sugars are moved into where they're moved into. Let's start that off then. So in summer, leaves make sugars, and these are loaded into phloem. So we are during the summer, we're explaining during the summer and then saying the advantage for the spring, aren't we? So we're telling this story of transport in the summer and then saying the advantage for the spring. That makes more sense. So how does this happen? This is the bit of theory that you need. So water potential falls because of this sugar. You put sugar in something, dissolves, and water potential falls, and so that will attract water by osmosis. So water absorbed by osmosis. Now we've got more volume of water, so we've got more pressure. So our hydrostatic pressure increases, and then the liquid will move from high to low pressure. So flow from high to low pressure. So we're just regurgitating the theory now. And the tubers, the little potatoes, the tubers convert the sugars, which will be sucrose, into starch and that will raise the water potential again. So water potential rises and water is lost. So it's just giving that theory of why the water's moving in at one end and why the water's moving out at the other end, and then you get a flow. A mass flow means all of the liquid all moves in the same direction. It's not like, diffusion or anything like that. And then the advantage for the tubers in spring, this allows growth using all of these wonderful sugars in the spring before there's a lot of energy from sunlight, so you can use these sugars for growth. So in spring, this allows growth using the sugars. And what we're making, we're using respiration, and we're making things like cellulose and so on. If I just put one of those, using the sugars in respiration. Or we could have another example, making cellulose, because that's what plants are made out of. So much more straightforward, and 5 marks. Crazy experiment to begin with.